In order to study how plasmalogens can protect neurons, the first thing you need to do is you need to create a system in which neurons are damaged or are going to become damaged. And to do that, scientists use animal models of neurodegeneration. I studied two separate types of neurodegeneration models. One model that focuses on white matter loss, like in multiple sclerosis or in stroke. And that model is called the cuprizone model of multiple sclerosis. Cuprizone is a copper chelating agent that, when ingested by animals, creates neural inflammation and results in white matter degradation, very similar to what we see in humans with multiple sclerosis. So when we take a look at that model, when we treat animals with plasmalogen precursors to elevate plasmalogens to protective levels, this whole concept of plasmalogen reserve that I've discussed in previous videos, when we generate plasmalogen reserve in animals prior to treating them with this white matter neurotoxin called cuprizone, the plasmalogen reserve protects these animals from neurodegeneration. So we can completely prevent myelin degeneration in a white matter model of neurodegeneration using plasmalogen precursors. The second type of neurodegeneration that we commonly study is called gray matter or neuronal degeneration. And to do that, we study using neurotoxins that specifically target specific neuron types. And one model in Parkinson's is called the MPTP model of Parkinson's. MPTP is a neurotoxin that was discovered in the 1980s from a group of kids who were experimenting with designer heroin, and one of the byproducts of their synthesis process was this MPTP. And all of a sudden, several people were admitted into emergency rooms showing symptoms of Parkinson's, and they were very young. And it was ultimately found that this cause of this was MPTP. And ever since then, we've used MPTP as a scientific model for Parkinson's. So very similar to the cuprizone model for white matter, the MPTP model for Parkinson's is if we treat animals with plasmalogen precursors in advance to create this plasmalogen reserve, and then subject the animals to this neurotoxin called MPTP, the plasmalogen reserve completely protects those animals from getting neurodegeneration, and it prevents those animals from getting Parkinson's. So it's very clear that plasmalogen reserve is neuroprotective in two separate independent models of neurodegeneration. We further studied plasmalogens as actual improving function in animals who already have experienced neurological decline. So in monkeys that have already been damaged with MPTP, so they have Parkinson's, if we treat those animals with plasmalogen precursors, we can improve the neurological function in these animals. So not only do plasmalogen precursors have neuroprotections, so if we can create this plasmalogen reserve, we can prevent animals from getting neurodegeneration. But even after neurodegeneration has happened, increasing the levels of plasmalogens can improve neurological function.